Hello everyone. In this lecture we're going to talk through some different modalities of inheritance, things that don't necessarily ascribe to the same rules that we have been previously discussing when we were talking about single gene and Mendelian inheritance. So we're calling this particular topic Non-Mendelian Genetics. So today we're going to talk about a few sort of major categories of different types of genetic traits. We're going to kind of look at these concepts of Mendelian, polygenic, and multifactorial. And then we're going to look at some examples of these traits in humans. So some that fit the polygenic pattern and some that fit the multifactorial pattern. And then we'll also talk about tools that we use to analyze multifactorial traits. So when it comes to our genes, there are very few genes that actually act alone. So the previous two lectures where we were talking about sort of what Mendel's work did and what he did with single gene inheritance, and in also the genetic inheritance topics of autosomal dominant and autosomal recessive conditions. Again, there weren't very many of those, so there's not a whole lot of traits that we know that act alone by themselves without any influence of other genes or other traits. So there are things that we know. We know that environmental factors and also other genes can modify the expression of a trait. So with that in mind, we can describe traits as follows. We can describe them as Mendelian, which a Mendelian trait is caused by a single gene. So we have looked at some of these before when we were talking about cystic fibrosis and Huntington's and sickle cell disease, traits like in your worksheet, we talked about having a widow's peak or having freckles, right? So those are traits that are caused by a single gene. We can also have traits that are caused by multiple genes. These are referred to as polygenic traits. So that is something that we see, for example, the pigment of your skin is one example of a polygenic trait. So you have multiple genes that are responsible for the melanin that is part of your skin tone. And then we have something called multifactorial traits. And multifactorial traits are where we have an interaction between genes and the environment. So even the trait that I just mentioned, skin tone, even though there are multiple genes that are responsible for it, and we could consider it polygenic, there is an impact of the environment also. So particularly for people who have lighter to fairer skin, if they spend time outside, you'll actually get more melanin production, and that changes the tone of the skin. So again, what we have here is we have these expression of traits that aren't always as simple as a single gene is responsible for this protein which causes this trait. More often than not, we have a lot of other things at play. So again, a polygenic trait is typically a trait that's responsible for, a trait that can be described as being due to the additive effect of multiple genes. So we have, again, that example that I gave you with the different genes for melanin production in your skin. There are 4 different genes, and so the more genes you have that has the allele for melanin production, the darker your skin will be, and the fewer of those alleles you have for melanin production, the lighter your skin will be. And in a polygenic trait, not all traits we see all genes have the same impact. So sometimes in a polygenic trait, there could be one gene that has a minor effect on the overall phenotype, and then there can be one gene that has a major effect on the phenotype. It just sort of depends on the particular trait. But what happens with a polygenic trait is we see this continuum of phenotypes. So previously, in all of our examples looking at Mendelian inheritance, you either had the trait or you didn't. There was no in between. So there isn't sort of a halfway point, if you will, for these traits. In polygenic traits, you can produce this continuum of phenotypes depending on the combination of alleles you have, which all add up. And again, they have this additive impact on the trait. So if you look at the individual genes themselves, they follow Mendel's laws in the sense that we have a dominant allele and a recessive allele, and the dominant allele can mask the recessive allele. But if you put them all together, they don't produce those Mendelian ratios. So we cannot use Punnett squares to predict crosses and say, oh, there's a 25% chance that this individual will have this trait and a 75% chance that this individual will have this trait. We can't do that with polygenic traits. They don't work that way. So let's take a look at one. And we're going to take a look at eye color here for a moment. And we're going to use, this is kind of an outdated model of eye color. It doesn't really work this way, but this is sort of the classic model that we use when it comes to a polygenic trait. We had this model that said there were two genes that were responsible for determining pigment of your eyes. The first gene, and we're going to use B for brown. Brown is dominant to blue, and blue is recessive. So we have gene 1, brown or blue. And then we also have gene 2, which gene 2 is capital G. And capital G is also dominant to blue, but capital G, green, is recessive to brown. So if you were to sort of put them in order, brown is dominant to green and blue, green is dominant to blue, and blue is completely recessive, if you want to look at it that way. So you could sort of put these all together, and what you would have is any combination of having a single brown allele would give you brown eyes. Okay. You have two combinations which would give green eyes, and then you would have essentially one combination which would give blue eyes. Again, this is kind of an outdated model. It's somewhat helpful when it comes to trying to give general descriptions of, eye color and how eye color gets passed on from generation to the next. But we think it's a lot more complex than that. We think there are actually 4 genes responsible. 2 of those genes produce colors called lipochromes, which are the blue-green, and 2 genes produce melanins. Melanins are brown. And there could potentially be more. We know, for example, that in mice, there are 60 different genes involved in eye color. So, again, there could very well be many more models here, but for the most part, this 2-gene model is sort of a kind of a generality of what we look for when we look at inheritance patterns of a polygenic trait. All right, so moving on. Let's talk about multifactorial traits. Multifactorial traits result from interactions of both your genes and the environment. And in a multifactorial trait, we do not have Mendelian ratios. That's why this is non-Mendelian genetics, because we can't use those old rules in order to figure out the probability of different phenotypes being expressed in different generations. So we can use this to predict phenotypes using a variety of things. So if we look at risk analysis, if we look at family history, if we look at how often the trait occurs in the population. So it can be somewhat of a predictive tool, but rather than giving us those straight ratios, 25%, 75%, 50%, whatever it is, it's more or less a likelihood. You are more likely to have this trait or less likely to have a different trait, but we don't follow those strict ratios. So all kinds of different multifactorial traits that we can talk about. We can talk about fingerprints and height and skin color and heart disease and diabetes and your risk of certain diseases like cancers, so on and so forth. And what we talked about just a minute ago was this idea of continuous variation. So continuous variation is used to sort of look at both polygenic and multifactorial traits, which many multifactorial traits are also sort of polygenic. So just to make that clear, you have multiple genes responsible for a trait, and then there isn't some impact of the environment as well. So they kind of go hand in hand a lot. And what we see with this continuous variation is you have a gradation in differences among individuals of population. So we use a standard bell curve to kind of describe this when we're looking at phenotypes. So if you've got a value of sort of the trait here, a lot of people will be somewhere here in the middle for that particular trait. There will be fewer individuals on either side of the bell curve, right? So we sort of have this mean, this average value of the trait. So the majority of people probably have this particular trait, and then on either end of it, they have a variation of it, and there are fewer individuals who have that particular variation. So let's go through some examples. The first one I'm going to do is fingerprint patterns real quick. This is multifactorial and polygenic. So there are a whole host of different ways to describe what your fingerprints look like. And if you've never done this, it's actually kind of fun. You can just take any color marker and just color a finger put it down on a piece of paper, and you can actually look and see what type of fingerprint you have. So we have different ways to describe the patterns here that we see. These are ridges, right? So these are the folds on your fingertips, and these are actually determined by, the number of ridges is determined by your genetics. So the number of those ridges, if we were to count them from top to bottom, that's a genetic trait. But the pattern itself is actually affected by prenatal touch. So during weeks 6 to 13 of development, when the developing fetus is touching the womb, that's where the pattern comes from. So even though the number of ridges is genetic, the pattern itself is actually caused by prenatal touch. So this is an example, again, of looking at sort of ridge count. So we're not looking at the pattern, but the number of ridges that students actually have. So this was a study that was done in a class, and they took a group of students, and they counted up how many ridges they had, and they plotted this. And again, you can kind of see this continuous variation pattern that I mentioned, this bell curve, right? So most of the class had something here in the middle. A few people had very few ridges, and there were a few people that had a lot of ridges, right? So again, we're following this bell-shaped curve. We can also look at this for a trait like height. So for example, the kind of interesting thing about this particular picture is this was done, like, I think 70 years apart from each other. So you had a group of, this is a group of all male cadets, as it were, and so they lined them up by height. And then this is a group of male and female students. And again, they were also lined up by height. And as you can see, again, we have sort of few people that were on the shorter end, few people that are on the very tall end, and everyone was sort of, the majority of people were here kind of in the middle, right? So once again, that bell-shaped curve that we talked about. And what's interesting is that over time, and again, over the, I believe, 60 or 70 years in between those two pictures, overall heights have increased. So we are taller on average as a population, as a human population, than we used to be. And a lot of it is really related to overall health and diet. So we are getting more nutrition, better nutrition at an earlier age and for a longer period of time. And that enables us to reach sort of a greater growth potential than if we were calorie deficient or if we were missing things from our diet. And there's been lots of different studies, something called a genome-wide association study. I'll talk about that in a little bit, that has identified all kinds of different genes associated with height. We can also look at body weight. So body weight is a multifactorial trait that, again, reflects energy balance. So how many calories you take in versus how many calories your body burns on a daily basis. And what we do know is that the trend in overall body weight has also been increasing. So all of these access to all of these calories that we didn't necessarily have 50, 60 years ago, we have an abundance of access to calories. And unfortunately, that is leading us also in a direction of becoming sort of a heavier population, which can have health consequences for the population as well. And scientists use kind of a measurement called the BMI, which is the body mass index. It is not a great, awesome way to determine different tiers of healthy weights. It's sort of the standard, but we also know that it does not work for everybody and every type, for example. This is just a sort of a graphic to represent how the prevalence of overall obesity, and obesity is defined as a body mass index greater than 30, how it's changed. And this is even since just the 1990s. And essentially the prevalence, again, is going from areas where we had a prevalence of, 20 to 30% of the population in most states. And then as we get down here all the way to, to 2020, which is right here, we can see that prevalence that has crept up to almost all states is now about 40 to 50%. And in the next 10 years, we expect in some places to see that prevalence to be over 60%. And this is representing what's referred to as severe obesity, which is a BMI over 35. So there are consequences, of course, to all of that. But here's the thing that we do know. There are genes that are absolutely responsible for body weight, in the sense that we know that there are genes related to things like hunger, for example, how quickly your cells can metabolize the energy that they've eaten, how hard your body holds on to, or how well it holds on to storage, extra storage for fats and things. There's a whole host of traits that are related to body weight. But the environmental part of this is, even though we have maybe potentially those predispositions, we also know that diet and exercise are environmental components that can impact weight. Heart health is another one. So just like the others, we have a whole host of different genes that can determine how well your blood clots, whether you have a high blood pressure, low blood pressure, cholesterol. All of those types of things have a genetic component. But we also know that your diet, your activity levels, whether you smoke, whether you exercise, can also impact heart health as well. So there's sort of a way that we can use sort of these studies, and we can determine how much of a trait is responsible with the relative contributions of genetics, and how much of a trait is responsible for the relative environmental impact. And again, we have those Mendelian traits would be on the far left-hand side of this particular graphic, and then things like being in a motor vehicle accident, there's no genetic correlation to that at all. That's purely just an effect of the environment. And as you can see, it's kind of a gradation. So there are certain genes that have a larger influence on certain conditions, like certain types of breast cancer and Alzheimer's risk. And then there are certain things that happen that are much more responsible when it comes to your environment. So smoking-induced lung cancer, for example. Another way that we can investigate multifactorial traits, and I'm just going to kind of quickly pull that one away. Another way that we can look at multifactorial traits is we can look at heritability. And heritability is where we are estimating the proportion of the phenotypic variation we see due to genetic differences, and how much of those are due to environmental differences. So again, we take these multifactorial polygenic traits and we try to determine heritability. And one way that we can look at heritability, we use something called twin studies, for example, which I'll tell you about in a minute. But the higher this number is, the more likely it is there is a strong genetic component. And the smaller this number is, the likelihood that there's a very low genetic component. So when you see heritabilities of 0.8, height, for example, verbal aptitude, 0.7, total fingerprint ridge count, 0.9. These are really, really high numbers. That means there's a really large genetic influence there. Numbers that are smaller means there's probably not a lot of genes here. So your mathematical aptitude, you likely did not inherit that. You likely developed that. So these are ways that we can sort of determine, again, how much of a trait is due to genetics and how much is due to environmental exposures. So one way that we can do this is we can use something called a twin study. Twin studies are really helpful because they tell us how much of a trait is probably genetic and how much is potentially due to their environmental conditions. So what we do is we take a series of different individuals. We can take adopted individuals, two people who share the same environment, but they have no genetic connection with each other whatsoever. We can also look at dizygotic and monozygotic twins. So dizygotic twins are just like siblings, any other pair of siblings. They share 50% of their genes, but they were raised in the same environment. And monozygotic twins have both an identical genotype. They share 100% of their genes, and they also share their environment. We can look at twins raised apart, who might share a genotype but not an environment. And then we do something called twin studies, and we look for something called concordance. And the concordance is measuring the frequency of the trait in both monozygotic and dizygotic twins. Twins who differ in a trait are said to be discordant for it. And for a trait that's largely determined by genetics, you have a higher concordance in monozygotic twins than dizygotic twins. What does that mean? It simply means that there's a larger genetic influence than there is an environmental influence. Okay, so here's concordance values. Okay, so again, the higher the concordance value in monozygotic twins, the more likely there's a genetic component here. So again, things like Alzheimer's disease really stick out. Autism really sticks out. Bipolar disorder, cleft lip, hypertension, schizophrenia. So all of these have much higher concordances for monozygotic twins than they do dizygotic twins, which means, again, there's an absolute large genetic connection here. I'm going to kind of skip over genome-wide associations. These are studies where we're looking at phenotypes in large groups of people. It's essentially a way that we can figure out if there's a genetic component to a disorder, for example. And we would find all the people who have it and compare them with people who don't, and then we would look at different pieces of their DNA to see if they match or they don't match. So this is the way that we try to figure out what genes are responsible for which particular conditions or disorders. I'm not going to go through the study design. We're going to skip all that. Here's a whole host of different ways that we have been able to look at different types of traits, cancers, allergies, and infectious diseases. These are a whole host of different genome-wide association studies that we're actually working on to figure out the genetic sort of backbone, if you will, of those diseases. All right, so that wraps things up. Again, the big takeaways here were the difference between multifactorial polygenic traits, how we determine heritability, and being able to give examples of all of those.